The overall goal of the following experiment is to quantitatively measure extravasation of circulating cells toward a chemokine gradient under conditions of physiological shear stress. This is achieved by first culturing a monolayer of endothelial cells onto collagen-coated microporous inserts and then assembling them into a flow chamber where they cover a separate static compartment containing chemokines. Then, shear stress is applied to the cells in order to mimic the physiological conditions in the vasculature. As a second step, test cells of interest, such as leukocytes, stem cells, or tumor cells, are allowed to circulate in the 3D system. During circulation, the contents in the static compartment influence the efficiency of interactions between circulating cells and endothelial cells, and subsequently the rate of transmigration of the test cells across the endothelial layer. Next, the 3D system is disassembled, and transmigrated test cells are collected from the lower static compartment in order to quantitatively measure cell extravasation using various state-of-the-art methods. Results are obtained that show the ability of circulating test cells to interact with endothelial cells under conditions of physiological flow and exit the circulation based on the concentration of chemokines in the static compartment.
The main advantage of this technique over existing methods like the use of parallel laminar flow chambers and static transfer assays is that it combines both shear stress in the upper compartment and chemokines in the lower static compartment. This allows researchers to quantitatively measure extravasation of circulating cells toward the chemokine gradient under conditions of physiological flow. This method can also be used to study the effect of a crosstalk between the cells of local microenvironment and endothelial cells on extravasation of circulating cells by adding an additional